there's almost limitless possibility here for next reactions. It's an exciting time to be a DEL chemist.
DELs, or DNA Encoded Libraries, are collections of millions of molecules, each tagged with a unique piece of DNA containing instructions on how to make it. These libraries are incredible resources for chemists, and Dr. Katelyn Billings, the Director of Discovery Chemistry at ZBAI, is in charge of making them. In this final Season 1 bonus episode of Bringing Chemistry to Life, we speak with another member of Chemical and Engineering News' 2019 Talented 12 about their work and trends in their field. I'm your host, Paolo Braiuca, Senior Manager of Global Market Development at Thermo Fisher Scientific. We began by asking Dr. Billings about the early experiences in chemistry that led her to where she is today.
I'm from northern Vermont originally, and growing up I was exposed to a lot of different potential fields of study. My mom was a chemistry major in college, while my dad was an English and journalism major. I found myself particularly drawn to STEM. I like the problem-solving aspect of it, and took pretty much every science class I could in high school. And chemistry just seemed to click for me, so I pursued my BS in chemistry at St. Michael's College in Vermont. And while there, I spent the summer after my freshman year actually doing physical chemistry research in my advisor's lab, and then I took part in the NSF's research experience for undergraduate program the following 2 summers, both in organic chemistry labs, one at UConn and the other at the University of Utah. And these experiences kind of helped me realize that I wanted to apply my chemistry knowledge and skills towards the synthesis of biologically interesting molecules. So I went to graduate school at Yale and joined Andrew Phillips' lab, where I initially worked on total synthesis of natural products. But when Andy moved our lab to the Broad Institute, I pivoted towards DNA encoded library design, synthesis, and evaluation. And during that time, I started attending DEL conferences, and this networking led me to join GSK as an investigator in their DEL chemistry group and eventually team leader. And then this past fall, I joined ZBAI Therapeutics as the director of discovery chemistry.
What happened to you during your PhD? You started with one project. You were doing total synthesis of natural compounds, which is very typical things for organic chemists, right? And then you moved to a different institute, and it wasn't necessarily your decision. Something had happened to you, and that's where you actually encountered the technology that has defined your career from that moment on. So there's a little bit of serendipity, actually. More than a little bit of serendipity in that. How did you ever think about it?
Yeah, definitely was serendipitous. I was admittedly unaware of DNA encoded library technology before our lab moved to the Broad. But I think I was always interested in learning about new areas of science and taking on new challenges. And DELs in particular I found fascinating because it gave me an opportunity to not only apply my synthesis skills, but I could learn about whole new areas of science, from combinatorial chemistry to target biology, assay design, informatics. And so really opened my eyes to a lot of new areas of science.
Can you tell us a bit more about ZBAI, what the company does and how it's different?
Yeah, so at ZBAI, we have a new take on DNA encoded libraries. So while others in the field are using it as a hit finding platform and making molecules directly out of the DNA encoded library as their hits, we're feeding all of the data that we generate in the DEL assays into machine learning models and then predicting the activity of molecules outside of the DEL. And so this lets us accelerate not only probe development and hit identification because we don't have to do the synthesis. We can predict the activity of commercially accessible molecules, but we can also leverage now all of the data from our assays to then accelerate our lead optimization efforts and get to candidate molecules even faster.
I feel like we should go back to this and discuss first about the DNA encoded library and let us understand what they are. With DNA encoded libraries, you certainly generate an immense amount of data, right? I would say this is true for any sort of library approaches. And the reality is that very few people spend any time in trying to extract meaningful information out of these experiments. And so it seems like ZBAI is quite innovative from that perspective. But, we really need to discuss about the library technology first. So why don't you start from there? Why don't you tell us what DEL is and how it works?
DNA Encoded Libraries, or DELs, are essentially large combinatorial chemistry libraries where each molecule in the library is attached to a unique DNA barcode that encodes its synthetic recipe. And the physical linkage of the small molecules to the DNA lets us screen billions of molecules all at once in a pool in an affinity assay against a target of interest, rather than needing to screen the library members individually. We're then able to PCR amplify and sequence the DNA barcode of the molecules that bound to the target to determine what the synthetic recipe was that was used to make them. And the field has really exploded in recent years, and now almost every major pharmaceutical company, as well as many biotechs and academic labs, are using DEL technology to either feed their drug discovery pipeline or to identify probes to help them understand fundamental target biology.
So you build up libraries. So I suppose you use some sort of combinatorial chemistry approach to generate diversity and explore chemical space. And you said you have these DNA tags that basically you use to recognize each individual molecule you build in your library, if I understand correctly. How does it work? Do you start with a number of starting building blocks and you attach a different tag for each of them? And then for each step of the reaction, you add another bit to the DNA, and then you grow this sort of barcode and unique identifier for each member of your library. Is that the way it works?
Yeah, that's a pretty good summary. So we start with a bifunctional molecule that you'll see called the headpiece a lot. And this is a molecule that at one end you have your chemical reaction handle, which is often an amine to start. And then at the other end you have double or single-stranded DNA onto which your growing DNA barcode is built. And so this material is aliquoted or split out into wells of a multi-well plate, and you attach a unique DNA tag. And then after a quick ethanol precipitation to clean up the ligations, you add a unique building block, which is then reacted to the headpiece, and add one building block to each well. And then once you have that covalently linked small molecule with your DNA, we can then pool all of the reactions together without losing any information, and then subject this material to global transformations or purifications, and then use that pool to then aliquot into a new well of a multi-well plate and proceed through the next cycle of ligations and chemical reactions. And then this will be repeated until the desired library is synthesized.
So every step of your synthesis is basically divided in two. On one hand, you've got the nucleic acid part when you grow your tag, and then you have the chemical part where you do whatever reaction you're doing to create your drug candidate, I would say, your API candidate or whatever that is, yeah?
Yes, exactly.
Okay. And I suppose that having a fairly complex multifunctional nucleic acid molecule there limits quite significantly the freedom that you have in your reaction conditions, right? So there's a limited amount of chemistry you can probably think of. Am I right?
I would say in a sense, so preserving the DNA tag is really critical, right? That's the only way that we're able to identify what molecules bound to our target. And so we want to avoid conditions that's going to degrade the DNA. So organometallic, strong oxidants, protic acids can be particularly problematic. But there's some other unique challenges as well. During my early graduate school training, when I was working on natural product total synthesis, we would often rigorously exclude water and oxygen from our reactions. But DEL chemistry is a totally different ballgame. For the DNA conjugates to be stable, you typically need at least 20% water in the reaction, which then limits the temperature range and some of the reagents that can be used. On-DNA reactions are also often run open to the air for practical reasons, so reactions have to tolerate oxygen, and then we're running reactions on the nanomole scale at sub-millimolar concentrations. We can overcome a lot of these hurdles simply by using a large excess of reagents. So it's not uncommon to see 40 to 1,000 equivalents of a particular reagent or building block, but because we're working on such small of a scale, the absolute amount of reagent is still quite small. Even with these restrictions, we're still able to generate diverse and interesting chemical space. The DEL-compatible reaction toolkit is expanding quite rapidly, but we're also able to take advantage of the fact that commercial vendors can provide building blocks that cover diverse chemical space, and they can leverage potentially non-DNA compatible chemistry for the synthesis of these building blocks, as long as then the end product has a functional handle that we can use in library synthesis.
Can you give us some examples of some go-to reactions you use frequently in synthesizing these libraries? I guess you cannot do lithiations, right? You can run our Grignard on these sort of things, but there might be plenty of other reactions that are still very useful.
Yeah, so the most commonly used reactions in DEL chemistry are those that we have large collections of commercially available building blocks that we can use to expand the chemical space that's covered by DELs. So you'll frequently see reactions involving amines, acids, aldehydes, aryl halides, and this lets us cover a lot of the workhorse reactions in medicinal chemistry, like palladium-mediated cross-couplings, Suzuki couplings, Buchwald-Hartwig, as well as amine capping chemistry. But there's been a huge growth lately in DNA-compatible reactions that either let us use new building block types or introduce new ways to connect building blocks using existing functional group handles. And we've seen CH activation chemistry, electrochemistry, and photoredox now all have DNA-compatible variants that have been developed.
It seems like you've got a lot of tools in your toolkit right there, so it's not as limited as I initially thought about it. How long and complex usually these syntheses are? Is there a certain limit, or is there a center point where this becomes too complicated because of the size of the molecule you are synthesizing there?
Yeah. Initially, the focus of the technology was on producing as many molecules as possible. So 4 cycles of building block introducing chemical reactions were quite common. But as the field shifts towards making more lead-like or drug-like molecules, 2 or 3 cycles of chemistry have become more common, so we can better control compound properties. I'll note that this doesn't take into account a potentially handful of steps that are used for non-diversity introducing reactions, like functional group manipulations or deprotections.
And do you characterize your molecules at all? Or do you just assume that something is happening and then you don't necessarily care, because when you run your testing, if something works, it just works, and then you're going to go back and look at it?
We do characterize our reaction products. LC-MS is the main means that we have at our disposal for characterizing our products, and what we look for is conversion from the mass of the starting material to the mass of the desired product. And if you see any unexpected masses, then you have to leverage your mechanistic understanding of the reaction to try and identify possible side products. We also use control molecules a lot. These can be helpful in understanding reaction outcomes, and we use a couple different types. So sometimes on DNA starting materials that can't undergo the desired transformation are tested to see if you have a change in the mass, then you know reactions are happening elsewhere on your DNA. We can also make standards off DNA and then attach them to use as reference for retention time if we're trying to determine selectivity if, say, regioisomers are possible. In library synthesis, in the first cycle of chemistry, the percent conversion to product of each reaction can be determined. But once we've pooled our first cycle of chemistry together, now we have a mixture of masses. And so instead, we're looking for bulk shifts in the distribution of mass. And this is where it's helpful to have prior knowledge of how well a building block behaves in a model system to understand how the system is performing. And in a similar vein, we can do purifications on the first cycle in library synthesis to remove side products. But after that, or if there's too many reactions in cycle 1, then is practical to purify, then instead you'll do a bulk HPLC purification on the material before continuing on to the next cycle. But here only excess building block or DNA tag is removed, but not specific side products. And so this is why you'll often hear that the DNA barcode encodes a synthetic recipe. Unreacted starting materials or side products can be carried through to the next cycle of chemistry and can either persist in the mixture or be competent in the next step. And any of those compounds could be what's binding to your target.
What is the advantage of DELS compared to other library screening technologies?
One of the great things about DEL technology is how accessible it is. With just 100 building blocks, a single chemist could make a library of 1 million products over 3 steps using split and pool chemistry. And while 1 million molecules is in the realm of an industry HTS collection, that would be unheard of on an academic scale if you had to individually buy or make each of these molecules. So DELs really lend themselves to being studied and applied across the board, whether you're in academia or in industry. But you can imagine in industry we have many more than 100 building blocks at our disposal, and so quite quickly we can make these large libraries and screen much broader chemical space than we could if we were making and screening individual molecules. And we can also run our reactions on much smaller scale, which then require us to use less material.
Right, so one of the key advantages is that you can run your test mixing basically the whole library, or maybe a significant part of your library together, and run it at extremely small concentration.
Yeah, we can screen billions of molecules all at once and get the information on that whole collection of our compound library in a single selection.
And this is something you absolutely cannot do with untagged libraries, because you need to do it one at a time, and that will require a lot of automation, big equipment. There's a lot of cost that certainly smaller groups or academic groups cannot afford, as you were saying. And is this due to the sensitivity of the tag? So because you only need a couple of really DNA molecules and you can amplify them with PCR, so you don't really need that high concentration. Is this the case?
Yeah, the molecular biology techniques are incredibly sensitive and powerful and let us go from single molecules to enough signal that we can then detect and sequence. And that gives us a ton of power there.
We hope you're enjoying this episode of Bringing Chemistry to Life. Stay tuned at the end of the episode for information on how to access content recommendations from our guest, as well as information on how to register for a free Bringing Chemistry to Life t-shirt. And now, back to our conversation. One of your research into expanding a little bit the scope of the chemistry of DELs, and I was reading your papers on the use of photoredox. That was actually very, very interesting, because at first sight, I thought that generating radicals when you have like a multifunctional DNA molecule in there wasn't such a great idea, but it seems that it works. Can you describe that work and what you found out and why it's important?
I will admit we were not convinced it was going to work either, and thought there was a good chance that the radicals would just damage the DNA. But the potential benefit of this type of chemistry was, I think, too large to not at least try. So one of the sets of reactions we were interested in was the sp2, sp3 cross-coupling of aryl halides with a variety of radical precursors. And generally how these reactions work is that upon irradiation with blue light, you have your photocatalyst, which can access an excited state. And this then leads to the generation of alkyl radicals through either an oxidative or reductive fragmentation of the radical precursor, depending on which building block type you're using. And then this newly formed alkyl radical enters a nickel catalytic cycle, where it likely adds to a nickel zero species. And then oxidative addition of the aryl halide to the resulting nickel 1 is followed by reductive elimination of our desired product. And then you have both catalytic cycles being closed by a single electron transfer reaction. And so the Molander Lab are experts in this type of photoredox chemistry, and so we pursued a collaboration with them. And working with James Phelan and Sharouk Badir and others in Gary's lab, we were able to show that acids, dihydropyridines derived from aldehyde feedstocks, aminomethylsilanes, and alkyl bromides were all able to be used as radical precursors. And we were able to demonstrate broad substrate compatibility, which is important to us from a library design perspective, since we want to be able to incorporate many diverse building blocks into our library synthesis. And you also can't optimize for specific pairs of reagents when you're working with pools of starting material. And we were also excited to see that our optimized conditions didn't damage the DNA, and we're able to show, importantly, that the products after the reaction could still have the DNA undergo successful ligations, could be PCR amplified and sequenced.
What kind of photocatalyst do you use? Just my curiosity. Is it some kind of iridium?
It depends on the radical precursor. So some we're able to use for CZIPN, which is an organic photocatalyst, and others needed an iridium photocatalyst in order to work.
And how do you go along developing these sort of things? I guess there are a couple of parts, right? You need to ensure that your reaction conditions are good enough to make the reaction product, and then in parallel you need to test the sensitivity of the DNA tag to the reaction conditions. So what takes prevalence? How does this work? Does it go in parallel, or do you prioritize one aspect or another?
They often go in parallel. We're able to get a sense of potential DNA damage when we're getting the LCMS of the reaction, so you can start to see losses of masses or different modifications that you're not expecting, and that can help guide your condition optimization. At the same time, we'll look to see, are we seeing high amounts of side products that we need to overcome by modifying the number of equivalents or adjusting the buffer that's used for solubilizing the DNA. That work is often done in parallel, and then we'll wrap up by looking at the extended DNA compatibility and put the reactions through a mock library scenario.
I just have one thing in mind. So once you synthesize your APIs, your potential biomolecules, bioactive molecules, you still have this sizable DNA tag attached to it. So when you run your binding experiment, don't you have the risk of interference from the DNA into whatever affinity is going to happen between the API and the biomolecule itself?
Yeah, you can get steric or electronic clashes between the biological target and the DNA if the DNA is too close to your small molecule. But to combat this, DEL chemists usually include an aliphatic linker between the two that extends the small molecule further away from the DNA to try to minimize the likelihood that this is going to occur, while at the same time not being so long that we alter the solubility of the DNA or its ability to undergo ethanol precipitation as part of the purification process.
What is next? What is the frontier here? What are the types of reactions that you are potentially interested in exploring, or where do you see more potential for the future for DELs?
I think there's almost limitless possibility here for next reactions, but some key areas that we're seeing popping up would be around enzymatic biocatalysis, looking at reactions that introduce selectivity, different stereocenters. Reactions with alcohols can be quite challenging because we're in an aqueous environment, and so getting selectivity there is key. Certainly additional development in the electrochemistry and CH activation space and photoredox chemistry. It's an exciting time to be a DEL chemist.
Indeed it is, and it's there aren't that many completely innovative areas in chemical research these days, isn't it? And this seems to be one of the few, which is really beautiful. It's very exciting. I like to jump back into what ZBAI does and the link between Dell's technologies and machine learning and artificial intelligence. So can you speak a bit more about that?
So Dell selection generates billions of data points, and machine learning really benefits from having a lot of data on which to train a machine learning model. And so it's really a perfect marriage of the two emerging technologies in its ability to impact drug discovery. What's exciting being at ZEBI, for traditional Dell synthesis, all of our molecules are made attached to DNA. And so if you get a hit out of a Dell library, you have to go back and synthesize the hit molecules off DNA. And this can be a quite time-consuming process, material-intensive. You're often limited in the number of series you might be able to follow up. By pairing it with machine learning, now we can take all that information and predict on virtual libraries that might be faster to follow up on, easier to synthesize in an accelerated time. We can apply them to libraries of molecules that we're working on throughout our pipeline. And so we really expand out the potential impact of the technology from not being just about hit identification and probe development, but now being able to impact the whole pipeline.
Now, as we got towards the end of our chat, there's a number of questions I'd like to ask you more related to your professional experience besides the scientific details. Most of our guests in the podcast are typically coming from academia. You made a precise choice of working in industry. You mentioned at the beginning that you were fascinated by how real the science is in industrial projects. Now that you have done it for a while, can you share with us what, in your view, are the pros and the cons of being a scientist in an industrial environment rather than being in a more free, let's put it this way, inverted comma, academic environment?
Like I said at the beginning, industry was always my end goal, and it came down to wanting to apply research to projects that would directly impact drug discovery. And in industry, we're often applying learnings from basic research to commercial problems on a larger scale or trying to solve specific problems. And it's more of a team-oriented environment. Publishing is less of a focus, but we're able to see the impact of our work on what our company produces. I find that to be enjoyable and exciting to see the real world impact of what I'm working on.
I suppose that the direction of your research is a lot dictated by market situations, the commercial opportunities of the company you work for. Do you struggle about this sort of lack of freedom? And do you feel like the direction is imposed to you rather than letting you free to explore areas, or are you still happy with the degree of freedom you have in your day to day?
I would say, commercial opportunities often define the big picture of what we're working on, but the execution of how we tackle the problem leaves open a lot of opportunity for creativity and freedom. So we talked about when I was at GSK, our challenge was to identify diverse starting points for drug discovery to feed into our pipeline. And one of the ways we chose to go after that was by developing new reactions for our Dell platform. But the specifics of how we went about that and what reactions we pursued was really driven by the creativity of our chemistry team. And so even within industry confines, there's still a lot of room for freedom of research, depending on the interests of the scientists that are working in different areas.
There's a question I always like to finish our interviews with, which is this. So you are still young, but you have already accomplished a lot. And I'm sure you have a bright future ahead of you and you're working on exciting things. So if you stop and think for a moment and you look back, what is one or two pieces of advice you would give to a young scientist just starting in their career?
I think what's really benefited me up until this point was reading the literature broadly, not just in my field, but related and adjacent fields, going to seminars, talking with different scientists, thinking about how they're approaching problems and ways that they're identifying to work on what's next. Networking is huge, and really not being afraid to pivot if you come across a new area of science that excites you, and being open in general to whatever happens or what you learn, and being open to different next steps than maybe ones that you had initially envisioned when you set off on your career.
That was Dr. Kate Billings, Director of Discovery Chemistry at ZBAI and one of the chemical and engineering news' Talented 12. Thanks for joining us for this final Season 1 bonus episode of Bringing Chemistry to Life, and keep an ear out for more. Season 2 is just around the corner. If you enjoyed this conversation, you're sure to enjoy Dr. Billings' book, video, podcast and other content recommendations. Look in the episode notes wherever you're listening to this podcast for a URL where you can access these recommendations and also register for a free Bringing Chemistry to Life t-shirt. This episode was produced by Matt Ferris, Matthew Stock and Emma Jean Weinstein.